We turn now to the form of neuroimaging which gets most popular press, which you're probably most familiar with from journal articles, from newspaper articles, from videos, and that's functional magnetic resonance imaging, or fMRI. The kind of images you see in the slide there should be familiar, and the language used to describe such images is often very florid, describing areas of the brain as if they were lighting up and so on. We'll need to be cautious as we approach this in order to understand what's going on. Functional magnetic resonance imaging uses the same equipment as we saw for the magnetic resonance imaging for MRI scans. So it's done in the scanner, but the signal that is captured is very, very different from that in MRI. In MRI, you recall, there were physical changes that were evident in soft tissue, which showed up as detailed anatomical images, recording the tissue throughout the entire head. The signal that's captured in fMRI is very, very different. It is a registration of a relative change in blood flow in one experimental condition compared to another. I said blood flow because the signal is a blood oxygen level dependent signal. So arterial and venous blood have different amounts of oxygen, and when, as we say informally, if one part of the body needs more blood, the heart provides more highly oxygenated blood. This is what's recorded in the fMRI. And having recorded this difference in regional blood flow between experimental conditions, we can then illustrate that graphically by overlaying it on a structural MRI, the kinds of images we saw before. Now, of course, the choice of color in those images is entirely a graphical choice. There's nothing colorful in the brain. In common with MRI, we have very, very good spatial resolution, and differences in regional blood flow in volumes as small as a cubic millimeter can be recorded. But we have very limited temporal resolution. It takes about 15 seconds to scan the whole brain in this way, and any change in regional blood flow due to something that happens in the environment, something that the subject undergoes, happens with about a 5-second lag. So we have basically no temporal resolution, but very good spatial resolution. And when I say we're measuring blood flow, it's important to remember that the signal that's being recorded here is not a signal of neural activity. The activity of the brain itself is not recorded. The brain is perfused with a very, very rich set of blood vessels. It is a metabolically very costly organ and is supplied very richly with blood, and you can see the blood vessels there. That's the basis of the signal that we get. So to recap, we start with an MRI image. That's an anatomical image of this sort. And then we record differences in regional blood flow in certain areas of the brain between two experimental conditions. So here, for example, we can see the so-called bold responses. Those areas shown in red and yellow are areas that had relatively more blood flow in one condition rather than another. They are not areas of the brain lighting up. The scan captures a volume and not just a single plane, so we can reconstruct these images in ways that are graphically very appealing with 3D models of the brain. And they lend themselves to perhaps over-ready interpretation. So how do you go about conducting an fMRI study? Well, you first have to decide what the contrasting experimental conditions are. And let's take a very simple case. Suppose we want to compare regional blood flow in the brain when someone has their eyes closed versus when they're looking at a picture of a kitten. So those would be our two conditions. The way we would go about this is we would first do a protracted high-resolution anatomical MRI scan. It takes about 15 minutes, but we need to know the precise anatomical details of the individual brain we're working with, because brains differ greatly anatomically from one person to the other. It's then necessary to do a lot of low-resolution scans monitoring the regional blood flow. Each scan takes about 5 seconds. Half of these will be done in one experimental condition, so with the eyes closed, for example, half of them in the other condition, for example, looking at a picture of a kitten. And it takes that many repetitions in order to get a useful signal. You then compare the two conditions, looking at one voxel at a time. Now, a voxel is a volumetric equivalent of the pixel in a picture. You're familiar with the pixel, the little dots that make up a digital picture. A voxel is a three-dimensional analog of this. So we compare the regional blood flow voxel by voxel across the two conditions. Now, the data that we obtain is always just of this sort. It's a comparison among two or more experimental conditions. That's the only data that comes from these studies, and it most emphatically is not mind reading. So these images are quite difficult to interpret. Here you can see a rather fuzzy structural average MRI, and you can see overlaid on this the regional blood flow increases that were associated with watching either a complex moving stimulus or a black screen, so very similar to the comparison we just said. You'll see that, unsurprisingly, there's an increase in regional blood flow towards the back of the brain here, which is where we located the primary visual centers. So, some caveats are necessary because these kinds of images have been often gravely misunderstood. The first thing to notice is that the nature of fMRI investigation requires a great deal of averaging. That is, we do something and we do it again and we do it again and we do it again. So some things that are not available here are the involvement of the brain in dealing with reality on an ongoing basis where everything is always new. Rather, if we show a picture of a kitten, show a picture of a black screen, show a picture of a kitten, show a picture of a black screen, and do this 150 times, we get the average of the activation in the two conditions and subtract. Now, I'm not sure what you can learn from this, but it is necessary to remember that whatever is going on is very static because it requires this averaging over time. And I'm fairly sure that from a subjective point of view, the viewer of the kitten the first time will interpret that picture differently from the viewer of the kitten, the same viewer, looking at the same picture for the 150th time. Another limitation of these kinds of studies is that, as I said, brains differ greatly in their fine structure from one individual to another. So averaging among subjects produces data that are difficult to interpret. Perhaps the biggest problem here, or danger that lurks, is one we have singled out before, which is the premature association of function with location. Remember, this was the phrenological mistake of the 19th century, which is that you project your idea of what the brain is doing onto a place in the brain. Such studies have often been criticized in this manner, and we saw examples of that. And a final caveat that needs to be remembered is that we are measuring blood flow, and the relationship between the activity of neurons and blood flow is not well understood. There's an awful lot of work still to be done here. So that when strong claims are made about the functional relevance of an increase in regional blood flow, it's difficult to know what that means in terms of interpreting the nervous system. Interpreting the nervous system is and remains hard. The immediately intuitive sort of appeal of these images is probably far more responsible for their prevalence in the public media than their informativeness. They are very, very difficult to interpret. They belong in a class of imaging techniques which have grown up in recent years based on very advanced physics, so complex equipment required producing signals that are difficult to interpret. Just to briefly mention another one, here's positron emission tomography, also done in a large scanner-like thing, but this time using radioisotopes in the bloodstream. PET, or positron emission tomography, also measures blood flow rather than neural activity, and so functional imaging of the same nature can be done in this way, but there are differences between them. They both measure blood flow. In a PET scan situation, you can allow a limited range of movement, but in fMRI, subjects have to be almost completely static, not allowed to move. We get poorer spatial resolution with PET, and it's very expensive. It's largely confined to, well, often used in clinical settings rather than pure research settings. On the business of fMRI, before we leave it, we've noted that the signal is difficult to interpret and that the subjects are restricted greatly in their movements. We also mentioned in the last video there's a huge interest in looking at the activity of brains when people engage in social interaction. Now, it's very, very difficult to study brains in social interaction when individuals are stuck inside a scanner. Let's just have a little look at a video from a Finnish research lab where they're literally trying to stuff two people into the scanner at once. This is Tommy Himberg.
Mutta sitten taas, jos me halutaan tietää siitä, että miten tämä hyvin rikas aisti-informaatio ja vaikkapa juuri ihmisten välinen kanssakäyminen tapahtuu aivoissa, niin meidän pitää päästä niistä yksinkertaisista malleista eteenpäin.
Olisiko toi ok? Painaako se? Poikkeuksellisesti pistämme sinne magneettikuvaukseen 2 ihmistä yhtä aikaa. Ja saadaan sitten sitä kautta tietoa, että miten esimerkiksi toisen ihmisen läsnäolo vaikuttaa meidän aivojen toimintaan.
Meidän tässä tutkimuksessa me ollaan asetettu koehenkilöt kasvot vastatusten, koska ihminen lukee toisen kasvoista paljon viestejä. Kasvot ovat tärkeä ärsyke vuorovaikutustilanteessa.
Eli meillä on jollain tavalla intuitiivisesti selvää se, että toisen ihmisen läsnäolo vaikuttaa siihen meidän olemiseen ja tekemiseen. Ja tässä sitten erilaisen koeasetelmin lähdetään selvittämään, että miten se näkyy aivojen tasolla.
Aivot ovat kuitenkin evoluution myötä mukautuneet nimenomaan tähän sosiaaliseen vuorovaikutukseen, sosiaaliseen ympäristöön. Eli siinä mielessä voisi ajatella, että siinä tutkitaan aivoja niin sanotusti omassa elementissään.
So, it's interesting that they aspire to more naturalistic conditions. There's something very, very unnatural, isn't there, about two people stuck in a scanner staring at each other's faces like that. So we've mentioned fMRI and PET scanning, and there's another related technology which is MEG, or magnetoencephalography. Just as we can record the electrical activity of the brain using EEG, we can record the magnetic fields that are associated with using MEG, which looks like this big hairdryer device stuck on top of the head. This provides excellent temporal resolution, as EEG does as well, with reasonable spatial resolution. Subjects can remain seated like this. There's even in that particular lab in Finland, we have multiple MEGs together so people can look at each other. So as we develop these new technologies, each with their own severe limitations, each one gives us a different form of information that we need to put together to combine to form a view of what the brain is and does. So there's a great deal of work at the moment in developing hybrid methods, combining EEG and fMRI. We saw NIRS combined with EEG, combining MEG with EEG, and so on and so on. So that's where a lot of activity is at the moment. And this is such a rapidly developing field that by the time you're watching this video, there'll be something new there as well.